Okay, this video is going to be glutamate brain damage revisited. I'm going to go through a lot of detail about glutamate. I'll make basic points that anybody can understand, but I am going to go into a little bit more advanced stuff. And that's one of the things that this channel offers is I'll go into more advanced detail, but I promise I'll make it so that anybody can follow that wants to understand glutamate. Okay, so anyways, we'll start out by just looking at the brain. The important point is this is the brain. There's what's called a tentorium. It separates the brainstem and the cerebellum from the brain above the tentorium. So this is called the supratentorial brain. And that's where your cerebral cortex is. Cortex means bark, like the bark of a tree in the periphery. If you had a tree stump in the periphery, that's the bark, that's the cortex. That's where the brain cells primarily are located. And the important point is 90% of the neurotransmitter in these neurons is glutamate. Glutamate is the on switch, activates neurons. GABA is the off switch. That's 5% of neurotransmitters. Then all the other famous ones are only about 1%. Dopamine, serotonin, norepinephrine, acetylcholine, all that other stuff. Okay. Okay, we are going to talk about this book a bit here. This is called Fat, Stressed, and Sick. We're going to get to this in about the middle of the talk. Author's name is Catherine Reed. She's a PhD biochemist. Her daughter had autism, and she was able to cure her daughter by removing glutamate from the diet. Okay, because you have too much glutamate, you can overactivate the brain. It's called excitotoxicity. It can cause anxiety, it can cause insomnia, it can cause loss of neurons. Even if you overact a neuron too much, that neuron can die. Okay, and I realize autism, is multifactorial in its causation. There's different subtypes. There's a whole spectrum, ASD, autism spectrum disorder, but it's just, it's good to know that some of them can be cured by withdrawing MSG from the diet. Here is this lady, Catherine Reed. She has a TED Talk. I'll put the link in the description below. She's very smart. It's a very good TED Talk. She also gave a nice interview. She was on Chef AJ. So I'll put the link to that as well. These are just some of the things that can be used to hide glutamate in the diet. What happened was the Japanese had figured out how to make food taste really good by adding MSG to it, monosodium glutamate. And it's really the glutamate. The body doesn't care about the monosodium. It only cares about the glutamate. So it can be manufactured free glutamate. It can just be glutamate. You can take any protein and just chop it up into individual amino acids, and that'll free up the glutamate and it'll become more available for eating. The proteins that have a lot of it is gluten. Gluten's in the ballpark of 25% of the amino acids, meaning every fourth amino acid is a glutamate. But there's a lot of it in corn, in whey protein, in soy. So when you process those proteins, you free up a lot of glutamic acid. Glutamate is sort of like deprotonated, but glutamic acid, glutamate, I mean all the same thing for our purposes when I use these words. And there's just a lot of chemicals that tend to have a lot of glutamate in them or related to them or contaminated with them. Okay, like citric acid. It sounds harmless, it sounds like something coming from citrus fruit, but it's not. Okay, we'll get into that later. I'll go through all the names for glutamate sources in the diet. This was a real popular book, came out about 1997, and this reviewed the history of the literature on glutamate and how it's an excitotoxin, meaning that it overstimulates the brain and can damage brain cells. Okay, here is sort of, you can divide this into 4 parts. There's actually more parts, and I'm going to go into them. I made a mnemonic for this. I was interested in I have the best theory in the whole world about neurodegeneration. It's, the Peter Rogers, MD. You can call it neurovascular uncoupling. A better name is supply and demand. I actually even have a third name for it. I'll call it the overworked and underpaid theory of brain damage. So what it basically means is you have a certain baseline metabolic rate in your neuron, and you need to supply it with oxygen and glucose. So OGD is oxygen and glucose delivery. Okay, that component of oxygen glucose delivery is a little bit like the Jack Dellatore theory, the vascular hypothesis of dementia. Other things that factor into it is how well can you run your membrane pumps of your individual neurons? How well can you produce energy in your neuron? How well is your mitochondria working? How well can you, speaking of pumps, how well can you get that calcium out of the cell? Calcium, so when glutamate hits a neuron, it activates it. Calcium channels open up, calcium comes into the neuron, then the high level of calcium in the cytoplasm gets the neuron to do whatever it does, like release its neurotransmitter. That's standard neuron communication. Okay, but then is you want to turn that neuron off very quickly, so you have to be able to pump that calcium out of the cytoplasm. You can pump it out of the cytoplasm into the extracellular matrix. I'll show you the pictures of all this in just a moment. The knockout exchanger, and you can pump it into the circa, into the endoplasmic reticulum. Okay, so I know some of these terms will be new to you, but we have to go through this. I'm going to show you pictures of all this stuff in a moment. It'll all gradually make more sense. But we are going to have to establish terms so we'll be able to have a dialogue of these things. Okay, mitochondrial inhibitors, you need mitochondria to make ATP. Excitotoxins are stimulants that can overactivate neurons. A classic one, of course, is glutamate, but there's other ones: beta amyloid protein, glyphosate, aspartame, quinolinic acid. Okay, we're going to talk about all these things. All right, now I'm going into a little more detail here. So this is sort of the same slide. I just added a little more to it. And by the way, this is a mnemonic. I made this up. POEM, P-O-E-M, okay? So P is for the pumps. You have to understand what's happening with the pumps. O is for OGD, oxygen glucose delivery. It's also for oxidative stress. Oxidants are things that steal electrons, like free radicals steal electron. They'll, for example, need an electron to complete their outer orbital. A free radical is a molecule that has an unpaired electron in its outer orbital, such that it needs an electron to complete that outer orbital, make an even number of electrons, complete the octet, for example, an 8-electron containing outer orbital. Okay, and we talked about excitotoxins, so P-O-E-M. I know I could have made it clockwise, but for whatever the reason, I just made it counterclockwise. It ended up working out better that way. Okay, and then mitochondrial inhibitors, and also microglia. We'll talk a little bit about microglia, how they are the macrophages of the brain, the main immune system cell of the brain, and they can become activated and cause damage. They can get activated by head trauma, excessive psychological stress. Infections are rare, but that can do it as well. But also these postprandial bacterial endotoxins, somebody's got leaky gut. So leaky gut can have a significant effect to cause brain damage. Leaky gut is no joke. Okay, now where does memory come from? The most important part of our memory center is in the hippocampus. So hippocampus, okay? And I joked, what university has the most fat people? The hippocampus. Okay, stupid joke, but the point is the hippocampus is the main memory center where you take in declarative memories, things that you can repeat, that you can say. There's different types of memory, like how do you ride a bike? It's not really something you talk about. That's procedural memory, and more of that, is in the cerebellum, for example. But standard declarative memory, you hear something and you repeat what you heard. That is largely the hippocampus, okay? So that's an important point. And the way you store a memory is you associate the new thing that you just learned with something you already know. That's how we map cognitive space. We associate the new thing with what we know. We put it into categories, okay, like classification categories, hierarchical categories. That's how we map cognitive space. Okay, now here is a sea squirt. Sea squirt, when it's young, it's like a tadpole. It swims around and has a brain. When it's an adult, it attaches to a rock and becomes a filter feeder. Its brain is reabsorbed. So you don't need a brain if you don't move. It's like the old question by Voltaire. He said, why do animals have brains but plants do not? Because animals move. As soon as you move, you have to make a value judgment. You have to choose a destination. I shall walk towards the fruit tree to get something to eat. I shall avoid that field over there because there's a bunch of coyotes. They might try to eat me. Okay, I'll stay away from that. Then you have to navigate to your destination. You have to avoid obstacles. You have to have a memory to remember to get back where you came from. Okay, so that's also why exercise, one of the main things that maintains your cognitive function and keeps you smart so you don't become fat, sick, and stupid like most late middle age and older people. It's totally avoidable. I see lots of really smart with it old people, and I see lots of, the vast majority of them are cognitively slow. Okay, here's 5 blind men and an elephant, just in terms of learning something. You first learn one little part. You don't want to jump to conclusion. The first blind man feels the tail, says it's a rope. Second blind man feels the ear, he thinks it's a rug. Next blind man feels the tusk, thinks it's a spear. The other one feels the side, it's a wall. Okay, one feels the leg, thinks it's a tree. So anyways, what we're going to do is kind of be like Aristotle said. Aristotle said the first step to an intelligent conversation is to remove emotions. So you don't commit. Just keep soaking up information, and then you'll have the whole big picture, and you'll be able to think intelligently about glutamate. Okay, and here I like this picture. This is Wanderer Above the Sea Fog by David Caspar Friedrich. And, it's called a Rückenförger in German, and basically the gist of it being that you're looking out, when you're looking at the character's back, you feel as if you are in the picture. And it's kind of a cool picture. The idea of it relating to the 6 blind men and the elephant is that you want to see all the parts of the elephant simultaneously such that you can then look down with a bird's eye view and you can make an objective assessment of what is going on and what is the best way to proceed. Okay, so that's what we're trying to do to get you to that point with regard to glutamate. Okay, and also, just so you know, no big surprise here, we're going to end up something similar to this quote. You've heard me say this many times. The healthiest way you could live is quite similar to Adam and Eve, except keep your indoor heating and plumbing. Okay, they're grounding their feet on the ground. They're getting some sunshine. They're getting some exercise. They're eating plant-based foods. Okay, when you look at the blood flow to the brain, it'll come up through the internal carotid artery. That's the anterior circulation. The vertebral arteries feeding the basilar are the posterior circulation, but anterior circulation is internal carotid artery, bifurcates. There's an anterior cerebral artery, which I won't show. That goes right down the middle. There is the middle cerebral artery, goes out to the side, and this is the money artery in the brain. It's the most important one. These little branches coming up into the basal ganglia here, basal ganglia level, are the lenticulostriates. Then you got the convexity branches coming up over the top, and they've got penetrating arteries that go into the deep white matter. At the level of the ventricles, it's called corona radiata. Above it is centrum semiovale. So the bottom line is this is how the brain is perfused. And where you run into trouble is if you've got hypertension and you overtreat it, then you can't adequately fill these arteries. So this is the top of the brain. Okay, it's the hardest spot to pump blood to against gravity. So when you eat a high-fat diet, it sticks the red blood cells together. It's like pumping a milkshake up there to the brain. Pressure has to go up, and then the hypertension damages the artery. Okay, if you just eat a normal plant-based diet, your blood stays the way it should be, normal, thin, like water, and your pressure doesn't go so high, and that's good. So you don't get a pressure damage effect on your arteries. Okay, that's an important point. Thick blood due to high-fat diets leads to hypertension, damages arteries. Also, if you have excess dietary sodium, that's a vasoconstrictor, and it clamps down on your arteries. Excessive dietary fructose can have a similar effect, not quite as severe, through uric acid being a vasoconstrictor. And vasoconstrictors can also lead to insulin resistance. We're not going to get into that right now, but they do. They contribute to, because you can't perfuse the muscle adequately, and thus you can't deliver glucose for glycogen storage adequately when you're vasoconstricted. Okay, here's a typical looking brain. Let's say this would be a typical looking brain in about an 85-year-old. All these white spots are little silent strokes. I mean, they might be symptomatic strokes, but these are associated with senior moments, okay? So I see tons of these all the time. You can see one patient can have hundreds of them. Let's just count a couple. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. You can easily see there's probably about at least 40 little strokes here, okay? I see this every single day. All right, now don't get me wrong. I see something even more common than this, okay? But these are people who are getting slow, walking slow, talking slow, thinking slow. Ain't what they used to be. All right, two main types of atherosclerosis. First type of atherosclerosis is Western atherosclerosis from high-fat diets. That tends to plug up the coronary arteries first, and the Johnson, okay? It'll also cause atherosclerosis in the internal carotid artery origin. By the way, you can look at the diameters of these arteries. Internal carotid arteries, about 6 millimeters in diameter. The coronary arteries, the big ones, epicardial ones, they're about 3 millimeters in diameter. The artery to the Johnson, the pudendal arteries, they're about 1.5 millimeters. So you just drop everything in half, 6 to 3 to 1.5. And so that's why the Johnson is often the first thing to present with impotence. And it's at least 30% of men in their 30s, 40% in their 40s, and at least 50% in their 50s are all impotent. And I can tell you it's super common. Again, if you just told me Western patient, I wouldn't even look at the chart, 60 years old, I would think pre-diabetic, diabetic, hypertensive, coronary artery disease, impotent, okay? Just because that's the vast majority of patients. By the time they hit late 50s, early 60s, they're fat, sick, and stupid. And it's totally optional. It doesn't have to be that way. And they got a fatty liver. Okay, they've often had a cataract surgery. All right, here's a quick Jack Delatorre theory. Bottom line is he tied off the internal carotid artery on one side in a mouse, and then about 2 months later, the mouse was demented. So all his middle-aged and older mice, you tie off their carotid in the neck, and they're demented. You do an autopsy on them. When he first did the autopsy, he expected a big stroke, but no stroke. It was just apoptotic. It was atrophic. That brain was shrunken on the side where he tied off the carotid. And again, people say, well, whoop-de-doo, how many people got an occluded carotid? Not that many. Yes, true, but lots of people have equivalent things. It's called the vascular hypothesis of dementia, whereby they get decreased cerebral perfusion, meaning decreased blood supply to the brain. That can be due to overtreated hypertension. If you take pills too much, you don't want that pressure too low. Remember what McDougall said, do not overtreat your hypertension. That's why he doesn't even treat until you at least have a systolic of 150, sustained for a long time at home, okay? He thinks almost all patients can get their blood pressure under reasonable control just with diet. It's rare, he thinks, for a patient to need antihypertensive medications. So what I'm trying to say is, if you overtreat that hypertension, you drive that pressure down too low. like he says, don't let the systolics, he likes to keep the systolic 85 and higher. You get that systolic down too low into the 70s and you drop that systolic too, you're not going to perfuse your brain. You're going to potentially get brain damage from underperfusion of the brain, hypoperfusion of the brain. A lot of patients got congestive heart failure. They're not perfusing their brains very well. A lot of them got atrial fibrillation. They're losing that atrial kick, 25% of filling. They might not perfuse their brain so well, especially if they're going at a rapid rate. It's a big mess. Okay, obstructive sleep apnea. They're hypoxic. Put a pulse ox on them. I've seen patients in their 60s for prolonged amounts of time at night. Overtreated hypertension. That, like I said, drops the pressure too low. Carotid stenosis, we talked about that. You can have aortic stenosis, aortic regurgitation. You can go for open heart surgery, CABGs, coronary artery bypass grafts. They keep them on the pump. They drop the pressure post-op too low because they don't want the patient bleeding out at the anastomosis. That's why a lot of patients have cognitive worsening after open heart surgery for coronary artery disease, because they were hypotensive both on the pump during the surgery and then in the post-operative course. The anesthesiologist run them hypo. They did that to my father, and he came out of it well, but my dad was a lot healthier than the average CABG patient. I wouldn't even have let him have a CABG if I had known more. That was like, over 25 years ago. I didn't know enough. I tried to talk him out of it, but I didn't know enough to force the issue back then. I was young. I didn't know all that I know now. Okay, and here's what I actually see most often. So here's a tale of two brains, okay? Here's a relatively normal-looking brain, let's say, for a 70-year-old. You've got the brain still uptight against the bone here. So I would call this a reasonably tight brain for a 70-year-old, okay? Whereas in this patient, you can see all this fluid right here. All this dark stuff is fluid, cerebral spinal fluid. You can see the brain has fallen away a good distance from the inner table of the skull on both sides. So this is diffuse cerebral atrophy. And that is what I see most common. Because I see many thousands of CAT scans and MRIs. This is a CAT scan of the brain for cognitive impairment, memory loss, dementia. And this is by far what I see. Just atrophic shrunken brains. Like the mouse, okay? But there's actually other roads that lead to that. Oh, this is just a seesaw drawing of, these are all the things that mess up your brain. We're going to talk about all these things, excitotoxins. A lot of these things are controllable, alcohol, exposure to these other toxins. We'll talk about, and here's all the things that are good for your brain. You can look at it more closely if you want. Here's how a neuron works. There is the presynaptic neuron over here, and here's the postsynaptic neuron. So here's the cell body where it has its nucleus, its DNA. Here's the dendrites for incoming information. The neurons are the cells in the brain that do the actual communicating. There's a lot of supporting cells. We'll talk about those in a moment, but the neurons are the ones that send and receive messages. So they receive information through the dendrites, then they send a message down their axon, and it'll reach the axon terminal, this part of the neuron. Then these neurotransmitters, NT is for neurotransmitter, will be transmitted across the synapse, which is the gap between the two neurons, and it'll bind with the postsynaptic neuron and activate it, okay? I'll show you another picture of this in more detail here. So enough incoming information activates this neuron and it fires an action potential. The action potential begins right here at the beginning of the axon. That's called the axon hillock. And then sodium channels open up. They depolarize the neuron, meaning making its inner charge more positive. So that wave of electrical charge is transmitted down the neuron until it reaches the axon terminal. Then it'll open up this calcium channel. This is called the VGCC, voltage-gated calcium channel. That's the VGCC, voltage-gated calcium channel. Calcium comes into the cytoplasm, and that activates the neurotransmitter vesicles to travel to the synaptic cleft and to release their neurotransmitter. The neurotransmitter will diffuse across the cleft, bind with the postsynaptic neuron, and then have an effect on it. I'm going through all this stuff so that when I start talking about the details about glutamate, everything will make sense. This is sort of like the background vocabulary. If you want, you can always hit the print screen button on your keyboard, and then you could make a copy of the slide or something to refer to later. Okay, what I'm talking to you is like the fundamental way to understand how the brain works. All right, so here's a neuron, here's the presynaptic neuron, here's the postsynaptic neuron. So presynaptic neuron in blue, postsynaptic neuron in purple. This right here is a supporting cell. This is an astrocyte. Astrocyte cells are called the mama cells of the brain because they help the neurons to communicate. So for example, the glutamate is a neurotransmitter here. The voltage-gated calcium channels open, calcium comes in, and then that activates the glutamate vesicles to travel to the synaptic cleft. They merge with the synaptic cleft. They release their glutamate neurotransmitter. It's called excitatosis. They release their neurotransmitter. It travels across the cleft. It binds to the postsynaptic receptors. This receptor here is a sodium channel. It's also called the AMPA receptor, but it's a sodium channel that lets sodium into the postsynaptic neuron. That causes a depolarization of the postsynaptic neuron, meaning it makes its inner charge more positive. When it becomes more positive, the magnesium is not so attracted to it. In this orange receptor channel for calcium, and the magnesium will pop out. Once magnesium pops out, then the calcium can go through it. Okay. Previously, the magnesium was attracted to the postsynaptic neuron because it had a negative charge inside of it before the sodium came in. But once it loses that negative charge, the magnesium pops out. But that's why magnesium also calms the neurons down. Remember, M for magnesium, M for mellow. So magnesium is a natural blocker of the NMDA calcium channel. Okay, and these are both glutamate receptors. Glu is for glutamate. So anyways, when calcium comes in here, it'll now activate the postsynaptic neuron. The glutamate then has to be cleared from the synaptic cleft. And the main way it's cleared is it's taken up into the astrocyte through these excitatory amino acid receptors. The glutamate goes into the astrocyte. It's converted to glutamine. And then the glutamine is sent outside the cell back to the presynaptic neuron. It has to be converted to something else. You can't be sending glutamate out here, or it could diffuse into the wrong area and start activating things. So glutamine is the safe way to transport glutamate without activating anything. Another little important point I wanted to show you here is that cortisol has a receptor on the synaptic terminal, and that will also stimulate the glutamate to be released. So remember, cortisol is elevated during psychological stress. So psychological stress increases glutamate transmission. And that's an important point. So anything psychologically stressed is going to be increasing activation of that neuron. And then you have to remember there's things that mimic psychological stress. They increase the same hormone. Caffeine will activate it. That's another reason why I never drink caffeine, not one drop. Sleep deprivation will also do it. So that's why you got to try to get your sleep, or you have the equivalent of being stressed. You're increasing activation of your neuron, and you don't want to do that, okay? Okay, a little bit just briefly on the pumps here. Here is the potassium-sodium pump, okay? And you've got the potassium coming into the cell. K is for kalium. That's the Latin word for potassium. Na is for natrium. That's the Latin word for sodium. One of the things you'll notice is in order to pump 3 sodiums out of the cell, remember you're pumping 3 positive charge out and you're only taking 2 positive in. So this effect of pumping more positive out than what's coming in, that causes the inner part of the cell to be negatively charged. So the negative charge inside the cell is negative 65 millivolts. All right. In order to do all this pumping, you need to use ATP. ATP is energy. It's like the $20 bill of the cell. So the ATP, in order to hang on to those phosphates, ATP means adenosine triphosphate, you need magnesium. It's got a positive charge that stabilizes the negative charge of the phosphates, so that the most external phosphate, the third one, is not released until appropriate for the pump's function. Okay, so that's another reason why you need magnesium to run all these pumps effectively. Magnesium is one of the most common dietary deficiencies. Forget about calcium. Nobody's calcium deficient, okay? Forget about protein deficient. Nobody in America is protein deficient. Nobody's deficient in fats. That's also totally exaggerated. Almost all Americans are deficient in things that come from plants. From fiber, they're deficient in potassium, they're deficient in magnesium, they're deficient in vasodilator type of nitrates coming from the plants. Okay, so if you are deficient in these things, you're not going to run your neurons that effectively. Okay, so that's part of why so many people are cognitively slow and not doing well cognitively. Also, fiber is needed to maintain the blood-brain barrier. We'll talk about that later in this talk. Okay, then this gradient now being established is an electrical gradient because you're pumping out so much sodium, and you got tons of these pumps, by the way. 1/2 to 2/3 of a neuron's energy goes to run this pump. In other cells, it could be as little as 1/3, but this is a super important thing. This is the electrical system of a cell. The energy generated from this electrical chemical gradient. So it's electrical because of the negative charge. It's a chemical gradient because you got more sodium concentrated outside the cell than inside the cell. About 10 to 1 is harvested by, you can allow sodiums to come into the cell and simultaneously use that to spin this thing like a wheel, and it'll pump. It'll be like a revolving sliding door to, let's say, like enter a hotel or something. This will then pump the calcium out. It's called the Naka exchanger. It's very fast. Here it is in more detail. The concentration of calcium is like 10,000 to 15,000 times higher outside the cell than it is inside the cell. And the point of that is, in order to pump calcium out of the cell, you're always pumping against a giant gradient. So you'll always need ATP to do that. And the fact that you always need ATP means you need good mitochondria. So again, here is our potassium, sodium ATP pump generating a concentration gradient because there's 10 times more sodium outside the cell than inside the cell. There's an electrical gradient because you're pumping more positive charge out than you are bringing in. And then you couple that electrical chemical gradient to pumping out calcium rapidly when you need to. There's other ways to pump calcium. I'll talk about those in a moment. But the bottom line is calcium is the aristocrat of ions. When calcium comes in, it starts bossing everyone around. Do this, do this, do this, do this, do this. It makes the cell do something. And the typical neuron, it makes it release its neurotransmitter. Whereas sodium, potassium, magnesium, they're the minimum wage workers, okay? They're doing all the work, but calcium gets all the glamour, all the glory. Okay, the secret of hypertension doctors don't know is that most people, our ancestors, let's say from a couple thousand years ago, even just from a couple hundred years ago, they ate like about 20 or 25 times more potassium than sodium, okay? Whereas we flipped it now. The typical American's eating about 10 or 15 times more sodium than they are potassium. And that's messing up their gradients, and that's also causing hypertension. And Richard Moore, the famous guy who spent his life researching ion pumps with regard to hypertension, he said, you need to eat a minimum of at least 5 times more potassium than sodium in order to avoid, you know, the more severe forms of hypertension. And that's very easy to do. Just eat more plants. P for plant, P for potassium. There's tons of potassium in plants. Okay, and the fact that nobody knows that about that potassium and nobody knows about hypertension being due to fat and lack of potassium right there and eating plants, tons of people die from hypertension, tons of them. Hypertension is a major killer. It's the #1 risk factor for atherosclerosis, #1 risk factor for stroke. You know, for atherosclerosis, that and cholesterol, high cholesterol. Okay, neuron cell body at rest. I'm just showing you that if you want to get this calcium out of here, you got to pump it. You can pump it into the endoplasmic reticulum. That's a calcium reservoir. The S for circa is just a carryover from skeletal muscle, so it's called sarcoplasm endoplasmic reticulum. Okay, but you don't even really need to worry about the word sarcoplasm in a neuron. Just remember the endoplasmic reticulum is a storage site for calcium, and this pump circa pumps calcium in there. It's a way to clear the cytoplasm out fast. And this is a great one right here. The knock exchanger pumps out thousands of them, 2,000 calcium ions per second, so it's super fast. You got this slower pump here, the PMCA, plasma membrane calcium ATPase. That one's much slower, but you know, it contributes, okay? All right, here's a little more detail about a synapse. And again, we're talking about a tripartite synapse. Tripartite means #1, presynaptic neuron, #2, postsynaptic neuron, and #3, the astrocyte, the mama cell. Okay, so again, either the voltage-gated calcium channels open up, causing the glutamates to travel to the synapse, or the cortisol comes in over here. And by the way, there's other things that increase cortisol. I forgot to mention them. MSG will actually cause increased cortisol. There's an entire book written about that. And these other causes of glutamate, you manufacture free glutamate. Excessive dietary sodium will cause increased activation of the sympathetic nervous system, which basically means the fight or flight response, which basically means it'll elevate cortisol. So excessive dietary sodium contributes to that effect. Okay, so all of these things are going to make the presynaptic neuron release its neurotransmitter. The neurotransmitter is glutamate. It comes across. It'll first bind the AMPA receptor. So this is AMPA R for receptor, and that's a sodium channel. Let sodium in. That'll depolarize the postsynaptic neuron, meaning make its charge more positive. When it becomes more positive, the magnesium is no longer attracted to the previously negative charge. It'll pop out of the synapse, and now calcium can run right through this NMDA receptor. So NMDA is the name of the receptor, R for receptor. The little S in front just means for synaptic, okay? There's other little minor points about it. We're not going to get into too much detail, but serine has a little bit of an activation component to this NMDA receptor. Calcium comes in, and under normal conditions, it actually increases BDNF in the postsynaptic neuron, and it does useful things. It's part of your memory. It's part of your normal activation of a neuron. If you have excessive EMF, like from holding a cell phone to your head, or you can get increased calcium entering this neuron. Okay, so you don't want to do that. All right, there's other little things get affected here. This is E is for extrasynaptic NMDA receptor. And that's also partly stimulated by glycine, and that's why it's thought that glyphosate, which is really, you can think of it as glycine phosphate, will also activate this NMDA receptor. Therefore, glyphosate is considered by the experts on the subject to be a excitotoxin, meaning that it's thought to increase the activation of this receptor, allowing more calcium into the postsynaptic neuron. That's bad. Aspartate will also do it. It's also an excitatory amino acid because aspartate is an amino acid. I call it baby glutamate because it's like glutamate, just one carbon shorter. But that's highly relevant, and that's the thing you get from aspartame. That will also increase calcium influx in an unwanted way into the postsynaptic neuron. So this E-NMDA receptor, E for extrasynaptic, because it's away from the synapse, this one is not supposed to be on very often. It should usually be turned off is the best way to think of it. These ones are much more frequently active. And so when you start overactivating this one on the side, you're getting too much calcium into this neuron, and it's going to start damaging this neuron. You can also activate the extrasynaptic NMDA receptors. They often call them MDARs, and get more calcium in there in a bad way. So traumatic brain injury can be very damaging to neurons. And when you get excessive calcium in there, it can activate things you don't want to activate. You can activate calpain, for example, which is a harmful protease enzyme that will damage the postsynaptic neuron. And its name is perfect. Cal for calcium activated, pain for painful consequences when that happens. Okay, we talked about glutamate being cleared from the synapse by the excitatory amino acid transporter on the astrocyte. And the glutamate comes into the postsynaptic neuron. It's converted into glutamine by glutamine synthase. Okay, so then it gets transported over here. All right, so we'll just kind of leave it at that. That's all we need to know at this point on that. Okay, here's just a little more detail. This is showing the microglia, and these can become activated. So you can even call this a four-part synapse when a microglia is being shown in the picture. And these can become activated, and they can do things you don't want to do. When the microglia are activated in what's called the M1 form, the bad activation form, they can even release glutamate and cause increased excitotoxicity. So excitotoxicity means too much glutamate overstimulating this postsynaptic neuron, and that can damage it. Okay, beta amyloid protein can also accumulate and stimulate these NMDA receptors and allow too much glutamate in there. Not good. Okay, I'll explain more about how beta amyloid protein can get involved, especially from a high-fat diet, for example. Other things, if you drop the blood supply of the neuron, you can get hypoglycemia, and that's also if you take too much insulin or something. Main point you can get from dropping the blood supply is hypoxia. Or if you have mitochondrial inhibitors, all of those things will drop the amount of ATP you can produce. You can't make enough energy. If you can't make enough energy, then you can't pump this calcium out. You can get overactivation of the neuron. So these are other roads, other pathways leading to excitotoxicity, meaning overactivation of the postsynaptic neuron. Okay, and the reason I'm going through all this, I realize this might seem too complicated, is I'm trying to give you the baseline understanding so when I start talking about glutamate, everything will make sense very quickly. As a matter of fact, I've gone through a lot of material, some of it rather complex, so I'm going to call this the end of part 1, and then I'll start up. It's probably going to be 3 or 4 parts. There's a lot of slides, but by the time I get done with this, glutamate will make sense to you, and you'll understand why you want to avoid it and how that will make you have a better chance to keep your brain cells and keep your smarts.